Good. Okay, well, I think I should start with probably an apology or two. Most of you will have understood from that that I am not a physicist, and I do not specialize in quantum mechanics at all or in quantum tunneling. But these are questions that are unavoidable, I think, to anybody who's interested in bioenergetics, and especially in the origin of life. So the question, I suppose, that has probably been the one theme that I've stuck with over 25, 30 years or so now, going back to my PhD on ischemia reperfusion injury, and yes, mitochondrial function, is chemiosmotic coupling, the idea that everything really in biology is driven by electrically charged membranes, by proton gradients across membranes. It's an enormously complex system, and it's very difficult to understand, well, how could such a system arise, evolve in the first place? What's the simplest possible version of it? And that's, I think, a question about the origin of life. And it's what actually most of my lab now works on. And so Hannah is with us, but she's done some of the work that I'll present. But what I'd like to try and do is give you an overview of the way that we are looking at the question, in the hope that some of the proper quantum physicists here will be able to tell us how we ought to be looking at some of these questions, or possibly be interested in collaborating on it. Now, the second apology I feel obliged to make at the beginning is this is a very particular view. I was asked, in fact, to talk about the origin of life, and a lot of people in the field would see the origin of life starting in terrestrial geothermal systems powered by UV radiation and involving cyanide, with that kind of chemistry. As a biochemist and as someone interested in the origins of metabolism, I have a lot of personal problems with those ideas. That doesn't mean they're wrong, it just means I have a problem. So I'm not going to talk about their work, but in terms of is quantum tunneling of any relevance to the origin of life? In my view of the origin of life, it absolutely is central to it. In other people's view, it's perhaps less important. I wouldn't really know. I can't talk for them. So the main question for me comes back to this idea of proton gradients across membranes. So usually about 3 orders of magnitude difference, proton concentration from one side of a cell membrane to the other for a bioenergetic membrane. This idea goes back to Peter Mitchell and Jennifer Moyle. Back in 1961 was the first paper on this, and it was in the 1960s that Mitchell was developing these ideas. Jennifer Moyle really did all the experiments. Without, she was a brilliant experimentalist. Without her experiments, Mitchell would have been forgotten long ago. Mitchell was a great thinker, cat-handed, I'm told, in the lab, and actually quite difficult to understand. It took a long time before his ideas were recognized. Probably very easy to understand for physicists, but for most biologists and biochemists at that time, he was speaking quite an alien language to them. There's a lovely quote that well, 2 quotes I'd like to bring your attention to here. One of them was from Leslie Orgel, who was a pioneer of origin of life work himself. And this was at the turn of the millennium, and he wrote in Science, "Not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, or Schrödinger." That's a really interesting claim to me. And the more you think about it, the more it seems to be true. If you think about the structure of DNA, for example, as soon as you see the double helix, you appreciate how everything works. It's just so logical, so simple. There's nothing counterintuitive about it. It explains everything. But the idea that cells would, rather than do things by chemistry, would have this intermediary of an electrically charged membrane didn't cross anybody's mind. Why on earth would cells work that way? And this is what I'll talk about. Now, Mitchell himself, I like this quote from him. From 1957 at a conference in Moscow on the origin of life. He said, "I cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So from the very beginning, he was thinking in terms, when he's talking about organisms, he's thinking about bacteria, single cells, critters with a membrane separating them, the inside to the outside world. And so it's just a single membrane that separates and links them to the outside world. It's not a common way of thinking about things, but when you think about the origin of life, it's a really powerful way of thinking about things. Now, most of the work that's been done since Mitchell's early ideas in the 1960s has really been on the actual nuts and bolts, the mechanics of chemiosmotic coupling. And that's what you can see here from a paper from Sazonov a few years ago. This is the respiratory chain in this is in fact in bacteria, in aerobic bacteria, but we have exactly the same thing in us. And I'll just talk you through it very quickly. So the gray bar across here is the membrane itself. And these colorful lumps are the proteins. This is complex 1 here. Across here is the ATP synthase. And this is complex 2, complex 3, and complex 4. Feel free to ask questions if you're not sure, but the only thing I really want you to take home from this is that there's a flow of electrons coming from NADH or from succinate, going into the respiratory chain, flowing to oxygen. Can you see my cursor? Yeah, great. So we have effectively a current of electrons from food to oxygen through this membrane. And that is powering the extrusion of protons, 4 protons at complex 1, none at complex 2, 4 at complex 3, and 2 at complex 4. It took decades. In fact, people are still working actively on these questions about exactly what is this stoichiometry, how many protons are pumped, how exactly are protons pumped, what exactly is the structure of these proteins, and so on. But this is roughly now the agreed stoichiometry for most organisms. And then it's the flow of protons back through the ATP synthase, which is driving the formation of ATP, the synthesis of ATP, which is generally called the energy currency of life. It's ATP, which is effectively driving protein function, changes in conformational state of proteins, all kinds of intermediary biochemistry, and so on. So without ATP, nothing is happening. And ATP synthesis is driven by the charge on the membrane, the proton gradient across this membrane, and the proton flux through the ATP synthase. Here's the ATP synthase. This is from John Walker, who got the Nobel Prize for this. In fact, only for the F1 head group here, the C ring embedded in the membrane itself, we still don't exactly know how that works even now. It's a rotary motor. And that came as really quite a shock, going back, in fact, to the 70s, lots of arguments in the 70s about how exactly it works. And in some sense, this is the most exquisitely beautiful enzyme mechanism that anybody has ever demonstrated. It's just gobsmackingly beautiful. And that's the best short animation of this that I've managed to come across. I suppose there's 2 problems with this. One problem is everything has it, pretty much. All of life depends on ATP and is chemiosmotic, which is to say it depends on membranes with a charge on the membrane and a proton or a sodium flux through the ATP synthase. So this is basically as universal as the genetic code itself. And yet it's a complex machine. Plainly, it's a product of natural selection and evolution. This is not something which pops into existence spontaneously as a result of chance. So unless God did it, and I don't think many of us would assume that God did it, then it's a product of natural selection, which means it's a product of genes, which means it's not linked with the origin of life itself. So how on earth did bioenergetics arise before the ATP synthase? This is the structure of complex 1, which is one of the proton pumps that I just showed you a moment ago. And this was a paper from Leo Sazonov from a month or 2 ago in Science, showing the structure of the proton extrusion mechanism of exactly what's going on. You can see it here in the kind of detail that I wouldn't expect anyone to understand. I certainly don't. But the point is, it's enormously complex. Again, this kind of proton pumping is not something that arose very early in evolution, and yet it's universally conserved across all of life. So there's a serious problem about how do we explain the universality of bioenergetics as we know it, given that it's basically an extremely complex and unexpected system. The other thing which is curious about complex 1 is within this matrix arm here. So this is the membrane down at the bottom here, and this is what's called the matrix arm. And you can just make out there's some iron-sulfur clusters, inorganic looking iron-sulfur clusters, 4Fe, 4S, a lot of them. And this is the structure of them. This is marking the distance from the center to the center, or from edge to edge is the one in brackets. And you can see that the distances are relatively short, 14 angstroms. These are in angstroms, these distances. So it's this kind of thing that has made people think about quantum tunneling in the respiratory chain. Now, the idea of quantum tunneling actually goes back to one of the pioneers of mitochondrial bioenergetics, Britton Chance, who was in Philadelphia for many years. In fact, Britton Chance was gold medalist at the Olympics in 1952 for sailing. He was the skipper of a three-man yacht. He published a paper in 1966 where he was working with the alga Chromatium. And this, in fact, was linked to photosynthesis. He was using lasers, and he was looking at the oxidation state of one of the cytochromes, which is the heme group. So using infrared spectroscopy, which, funnily enough, I was using in my PhD as well, based on Chance's work. And he showed that taking the temperature down to 100 Kelvin, in other words, limiting the activation energy, that there was very little relationship between the activation energy and the speed of electron transfer, which he ascribed to tunneling from the cytochrome through to, from the chlorophyll through to the cytochrome. That seems to be showing it the other way. I think it should be going that way. But anyway, the point is, very beautiful, very simple experiment, simply showing that in the cold, the rate of electron transfer is almost exactly the same as it is in warmer temperatures. And he proposed that it could only happen with quantum tunneling. So that was the first year, to my knowledge, that is being proposed in biology. 1966 was actually a remarkably good year. I'll touch on one or two of these. The reverse Krebs cycle, you may be familiar with the Krebs cycle in mitochondria. Effectively, what it does is it takes food, it strips out hydrogen and CO2, passes the hydrogen into the mitochondria where it's burnt with oxygen to generate ATP via this machinery I've shown you. So the Krebs cycle is producing CO2 and hydrogen and generating ATP. The reverse Krebs cycle is doing exactly the opposite. It's taking CO2, it's taking hydrogen, it's taking ATP, or an energy currency, and it's making organic molecules from it. And this is very tightly linked probably to the origin of life. I won't say much more about that now, but it's very linked to where on earth organic molecules came from in the first place. Eck and Dayhoff, they published a classic paper in 1966 on ferredoxin, which I'll talk more about in a little while. And Peter Mitchell, who I've already mentioned, his little grey book was also published in 1966. I was born, incidentally, in 1967, so things went downhill pretty quickly from there. Probably the person I associate who's done most work on quantum tunneling in the respiratory chain is Les Dutton, and I'm sure a few of you will know him. And here is one of the first figures he had in a Nature paper back in the 90s. And this is showing the reduction potential in volts. And you can see that there are various endogonic reaction steps here, which are made possible by the distance, by relatively short distances, increase the likelihood of a quantum tunneling event. And this was from one of his papers. My understanding is that effectively 1 angstrom additional distance slows down the rate of electron transfer by about 10-fold. So it's very, very sensitive to the distances involved. And generally, to have very fast electron transfer at less than 1 millisecond, you need to have distances of less than about 7 angstroms. Distances of up to about 14 seem to work. Beyond that, you're unlikely, is my understanding, to have much in the way of quantum tunneling. And these are, again, from one of Les Dutton's papers. These are the distances in complex 1 between the various iron-sulfur clusters, complex 2, complex 3, complex 4. I won't go through them in detail. The gist of his argument is that it's really distance that has been selected for in biology more than anything else. In fact, he had this lovely title for one of his earlier papers. Darwin at the molecular scale was the title of one of his papers. And it was that paper that really grabbed my attention as something which, I have to say, when I first came across it, I was astonished that selection would be operating on distance in this way and tunneling in that way. I should have known more about it. That was the first time I came across it. And I've asked various bioenergeticists since then, because I initially thought that this was pretty out there work. And I was quite surprised to find that pretty much all the bioenergeticists whose work I respect a great deal, like John Walker, all immediately says, yes, this is bona fide. It's very well established, which is why I started to take it so seriously myself. This is slightly more recent work, and this is something that perhaps some of you can help me with. The way that I would interpret this is merging of resonance states. So something as a simple biochemist, I would interpret this as being a little bit like electron delocalization in a benzene ring or in a carboxylic acid or something, which is to say, if an electron enters a respiratory chain, there's a certain probability that it could be anywhere in that chain. Which must make it, I would assume, and perhaps you can correct me on this, easier to reduce any of these iron-sulfur clusters if you cannot localize the electron to any specific cluster. If there's a certain probability that it might be at a different cluster, that must make it easier to reduce. That, I assume, and again, I'm speculating here, would allow you to tune the reduction potential, not of the cluster itself, but of the entire chain. And I don't think that anybody has ever given a satisfactory answer as to why there are so many iron-sulfur clusters in complex 1, for example. Why is it that there are 9 of them there? It doesn't seem to be simply a case of you have to cross a certain distance. There's nothing that would prevent NADH from binding down at this end of the chain rather than at the other end of the chain. There's nothing in principle that would prevent that happening. And yet this structure has been universally conserved. So there seems to be a tuning here, which is rarely discussed, about the number of clusters involved in these chains. And I hope that that's something that we can talk about later on when in the discussion. Now, I'd like to take a few steps back and come at this from the point of view of the origin of life, where things cannot possibly have been so complicated. And again, I'm going to go back to Peter Mitchell and to the questions that he was originally interested in. And they were actually remarkably simple questions, and I've simplified it for myself even more. The question really was, how do bacteria keep their inside different to the outside? How do you not just become the environment? How do you avoid equilibrium with the environment? And it's obvious that you're going to have to pump things out or bring things in. That's a statement of the obvious. And it seems surprising to me, but Mitchell appears to have been one of the first people to realize that these membrane proteins are going to have to be effectively as exquisitely sensitive to their substrate as enzymes are. In fact, they are basically enzymes that sit in the membrane. I would have thought that this is kind of common sense, but apparently back in the 60s, it was by no means common sense. So you need to recognize a proton, and you need to pump out a proton in this particular case. And it's going to cost energy if you wish to have more protons outside than inside. So that was all the statement of the obvious. What Mitchell realized was that if you simply allow that to reverse and you allow the protons to flow in, then in principle, it will be possible to tap the energy from the influx of protons. And the mental leap here was going from bacteria trying to keep their insides different to their outside, to thinking about respiration in humans, to realize that the missing link that people have been searching for in terms of chemistry for decades at that point was in fact not a chemical intermediate, but an ion gradient across a membrane. So that was the leap. And really what he, I think, prided himself on as an idea, more than the nuts and bolts of chemiosmotic coupling, was the idea of vectorial chemistry, that cells are not just a bag full of enzymes that do chemistry, but a lot of the activity is happening across the membrane involves charges across the membrane, charges on the membrane, and has a direction in space as well as time. So it's not simply scalar chemistry inside a bag. Now, again, it seems like a very simple realization, but it really transformed the way that people thought about bioenergetics. And I think still, as a field, the origin of life is not thinking in terms of vectorial chemistry, in terms of cells having a membrane and all the activity is across the membrane. So bacteria are powered by letting the outside in, effectively by allowing protons to flow in or sodium ions to flow into the cell from the outside. And it's important to realize that this energy flow is powering growth, which is to say carbon metabolism to turn CO2 and hydrogen into organic molecules, Krebs cycle intermediates, amino acids, nucleotides, and so on, and energy metabolism. So ATP synthesis is the simplest way to think about that. And plainly, it costs energy to pump these protons and sodium ions back out again. And there's been this long question, chicken and egg question, really, which came first and why, and what happened at the origin of life. And there's been a tendency to assume that all of this machinery is so complex that it must have arisen relatively late, even though it's universally conserved across all of life, and that you'll have to pump things out before they can come in. Well, that really does an injustice to the environment, to the power of the environment that Mitchell invoked from the very beginning. So how can we begin to get at it? Well, phylogenetics has transformed the way that we think about these questions in biology over the last 20 years or so. And this is from Bill Martin. This was actually a conceptual tree from 20 years ago that Bill Martin put together. And there's a couple of things I'd like you to notice about it. There are 3 domains of life. So forget about plants and animals and fungi and so on. We are all eukaryotes and we're quite closely related to each other. We are very closely related to mushrooms when it comes to this kind of phylogenetic tree of life. Then there's the domain of the archaea, which look like this, and the bacteria, which look exactly the same. The archaea were discovered first in the 1970s, late 1970s, and recognized as a separate domain of life by Carl Woese in about 1990. And there's a couple of things I'd like you to notice about this tree, because this was radical, really radical when it was put forward. And it's now been, I would say this is now close to the recognized truth in evolutionary biology, which is a remarkable statement, really. So one thing is here, the eukaryotes, all complex cells derived from an endosymbiosis, apparently a singular endosymbiosis between an archaeal host cell and a bacterial endosymbiont, which went on to become the mitochondria. You'll see there's another endosymbiosis here with the chloroplasts, which gave rise to algae and then plants. But that's later on and only applies to those groups, not to all the eukaryotes, not to fungi, not to animals, and so on. So what that means is that the eukaryotes are a derived domain. They're not a primary domain of life. They are derived from the bacteria and the archaea, and therefore they are not relevant to the origin of life. So I'm going to say no more about them today, except we can talk about them later on if you're interested. The second thing, which is really radical to notice here, is that he has Luca, this is the last universal common ancestor of all of life, emerging from, well, inside a hydrothermal vent. And you can see the bacteria are coming separately out of this vent to the archaea. Now, I saw Bill give a talk on this back in 2002, and he's quite a brash Texan, and he was more or less trembling before he gave the talk because he was very uncertain how this was going to go over and whether he was going to kill his own career in these radical ideas. And I have to say, when I saw his talk, I thought the guy is completely nuts. And you know, I now think that he's bang on, and that this was really radical thinking. And it took me, but I think most of the field, a long time to catch up with that place. So here's why he argues that bacteria and archaea emerged separately from a common ancestor that lived in these vents. So we know they had a common ancestor. Because this is a gross simplification, but bacteria and archaea share a great deal in common. They share the universal genetic code. It's exactly the same code in bacteria and archaea. It's rather a strange code, and the chances of it evolving by convergent evolution would be negligible. We also share, bacteria and archaea share the ribosome structure. These are the protein building factories. Most of the enzymes involved in transcription and translation. So this is the processing of DNA to make RNA and then converting RNA into proteins. Quite a lot of intermediary biochemistry, bits and pieces of the Krebs cycle, and membrane bioenergetics. Both groups are obligately chemiosmotic, which is to say they depend on charging their membrane in this Peter Mitchell mechanism. But they're fundamentally different in other respects. The cell membrane is the major one to take notice of here. The membrane of bacteria and archaea is not homologous. In terms of their biophysical properties, they're quite similar to each other, but in terms of their actual chemistry, including the stereochemistry, they are not the same. And so they appear, arguably, to have had independent origins. That also goes for most of the lipid synthesis enzymes. It goes as well for the cell wall. It goes for central pathways in metabolism, like glycolysis, which you would have thought would be ancient. Well, the pathway seems to be ancient, but the enzymes that are catalyzing it are not the same, not homologous in bacteria and archaea. Remarkably, DNA replication. There's about 200 enzymes involved in DNA replication, and only about 2 of them are homologous in bacteria and archaea. And the respiratory chains, which are pumping protons, again, this is more problematic because there's been a lot of lateral gene transfer, but they don't appear to be homologous in bacteria and archaea. So there's a deep divergence between these 2 fundamental groups in life. And so there's a nice paradox, which is that membrane bioenergetics are universal across life, but the membranes themselves are not. And now this paradox could be resolved if life originated in an environment where there were geologically sustained proton gradients. Is there such a place? Yes, there is. This was discovered by Deb Kelly, who was captaining the Alvin submersible back in the year 2000. They were actually on an expedition to the mid-Atlantic ridge. And before they got to the ridge, they were about 20 miles or so away from the ridge itself. And a PhD student was apparently looking out of the window of the submersible and noticed this magnificent hydrothermal vent field. Nobody expected there to be anything there. The ocean floor is supposed to be a desert. When you get to the mid-Atlantic ridge, then you start seeing black smoke events and all kinds of interesting things. But this was not supposed to be anything here. Well, it turned out that this lost city vent field, you can see just from looking at it that they don't appear to be active. There's no black smoke belching out of these vents. In fact, they are active. The whiter bits that you can just make out here and on this bit, they are actively venting, just not smoke, as you're perhaps more familiar with. These are known as alkaline hydrothermal vents, and they're produced by a reaction between olivine, which is a mineral which makes up a large part of the mantle of the Earth, and water, which will percolate down into the rocks beneath the seafloor, react with it, metamorphose it into serpentinite, releasing hydrothermal fluids which bubble back up to the base of the ocean and precipitate into these vent structures. Now, these vents had been predicted to exist 10 years before they were discovered by Mike Russell. And again, he's another person whose work has influenced me enormously. And he was talking about proton gradients across barriers in alkaline hydrothermal vents throughout the 1990s, and nobody listened to him until the discovery of Lost City, at which point suddenly his ideas became quite mainstream. And this, remarkably, was a feature article run by Nature, where they described him as Naissance Man, as in the birth of life, punning on Renaissance man and dressing him up as Erasmus, the Renaissance philosopher. And here he is at JPL with his high pressure reactor, trying to simulate the conditions in these vents, and a little bit of Lost City hydrothermal system behind. On the right-hand side of this is an indication of the structure of these vents. They are basically microporous labyrinths of interconnected pores. And Mike really imagined that these were electrochemical flow reactors where you have reactive fluids. So the hydrothermal fluids are alkaline, They're rich in hydrogen gas, I mean, practically bubbling in hydrogen gas, some methane as well. And ocean fluids percolating in, much richer in CO2, iron. There's some sulfide coming out of the alkaline hydrothermal system. So you would expect to see iron sulfide minerals precipitating in these vent walls. So you have catalytic walls with reactive fluids at temperatures of about 70 degrees centigrade. So it's a beautiful conception of an electrochemical flow reactor for the origin of life. And Mike has been developing these ideas for 30 years now. There was a 30-year anniversary of his first paper in Granada last year, where a bunch of us went. And they, I won't say they're mainstream yet, but they're one of the more serious ideas in the field. They're not mainstream insofar as I mentioned at the beginning, the people working on terrestrial geothermal systems and cyanide chemistry are probably the dominant voice in the field, but this is an important voice, which is, I think, more appreciated by microbiologists and geologists, and probably physicists as well, who are interested in the origin of life, much less so by chemists. Who are interested more in the yield and the product range that you might actually get. So for a long time, the problem with this work was that there wasn't much good chemistry supporting it. That's changed in the last few years. So here is the context, I think, as originally put forward by Mike Russell and as we imagined it in a paper a few years ago. So here's what I showed you already. This is a bacterial cell, imagine, with a membrane pump pumping out protons. So we have a difference in proton concentration of about 3 pH units, 3 orders of magnitude between the outside and the inside of the cell. Of course, there's buffers and so on in modern systems, so it's not just simply a 3 pH unit difference, but that's roughly what it equates to. And here we have an inorganic pore in a hydrothermal vent. This is massively thicker than a cell membrane, but the polarity, which is to say acid outside, alkaline inside, and the magnitude, which is to say about maybe 3, maybe more than that pH units between the inside and the outside, it could be anything up to 6 pH units, are rather similar. Now, that doesn't mean anything. That's simply analogous, but it is a potential starting point, and it does allow us to start formulating some hypotheses about how things might have happened. There is a second point, which I mentioned briefly already, which is that these walls in these alkaline, in the pores in alkaline hydrothermal vents would contain iron-sulfur minerals back in the Hadean. Not today, not in modern vents, because the oceans are full of oxygen now. But in the Hadean, when there was no oxygen in the atmosphere, the oceans were ferruginous, which is to say rich in iron, dissolved iron, and there should have been iron sulfide minerals precipitating in these walls. The proteins which are doing this pumping and so on are very often iron sulfur proteins, and they contain iron sulfur clusters, as in ferredoxin here, which have a structure. This is the structure of a 4Fe, 4S cluster, 2Fe, 2S. You see them both in ferredoxin here. It's basically very, very similar structure to the Greigite half cell. So you can imagine at least that there's some kind of progression between the minerals that you see in the walls of hydrothermal vents and the iron sulfur proteins that are still reducing CO2 to make organic molecules today. This idea of the antiquity of ferredoxin was put forward very strongly in a beautiful paper from Margaret Dayhoff back in 1966. I mentioned it was a good year, 1966. I still find this paper gives me spine tingles when I read it. She looked at the sequence. This was very, very early days of sequencing. She looked at the sequence. She came up with this modern code of a single letter for an amino acid. It was all the 3-letter code back in those days, but it was easier to work with single letters. And she found a repeating sequence. I won't go through this in any detail. This is from the abstract. That it evolved by doubling a shorter protein, which may have contained only 8 of the simplest amino acids. And this shorter ancestor in turn developed from a repeating sequence of the amino acids alanine, aspartic acid or proline, serine and glycine. So just 4 amino acids and some of the very simplest, earliest ones, which would be this repeating pattern here. And she says it suggests the incorporation of its prototype into metabolism. So this is a prototype ferredoxin into metabolism very early during biochemical evolution, even before the complete modern genetic code existed. So this is a thrilling conception of how life might have started and places ferredoxins right at the very heart of it all. So what do we know now? That was back in 1966. Well, I mentioned these two groups, the bacteria and the archaea, the two domains of life, the primary domains. They have, as I said, surprisingly little in common. And there's only one pathway that they both share of CO2 fixation, which is called the acetyl-CoA pathway. And both of them start with CO2 and hydrogen. And methanogens, which are archaea, make methane as a waste product. And acetogens, which are bacteria here, produce acetic acid or vinegar acetate as their waste product. And the overall reaction in both cases, the delta G naught prime, is exergonic. So overall, it's releasing enough energy to drive some biochemistry. So this is the only CO2 fixation pathway found both in bacteria and archaea. It's reacting hydrogen with CO2 to make organic molecules. Hydrogen is exactly what you get bubbling out of the ground in these vent systems, and the early oceans were rich in CO2. Basically, every pathway, every serious pathway across all of life that is, which is incorporating carbon into biochemistry, is fixing CO2. There are other ways of doing it, sometimes CO, never cyanide to my knowledge, or at least certainly no major branches of life are doing that. So it's really CO2 was there from the beginning in the atmosphere and is there from the beginning in phylogenetic pathways and so on. Now, under alkaline hydrothermal conditions, the reaction between CO2 and hydrogen to make total cell biomass is exergonic overall. So this is a theoretical thermodynamics study from Jan Ammond. And you can see here, this is the delta G, negative numbers as we come down. This is total cell biomass. This is the temperature on the x-axis. So this is roughly the temperature of alkaline hydrothermal vents. They're not that hot, they're warm rather than hot. So around about 50 degrees centigrade is quite strongly exergonic to produce total cell biomass from CO2 and hydrogen. Again, we know that must be true, otherwise methanogens and acetogens couldn't possibly exist. They couldn't grow from hydrogen and CO2 if it was not exergonic overall. But it's still surprising to me that if you were to take a test tube, fill it with hydrogen and CO2 and put a catalyst in, thermodynamics says you'll get cells coming out. Of course, we know you don't get cells coming out, but that's what thermodynamics alone tells you. Mostly amino acids, quite a lot of fatty acids, saccharides, sugars, that's to say, is fairly close to zero, and nucleotides cost energy to make nucleotides. But if you can conserve some of the energy released from amino acid synthesis and put it into nucleotide synthesis, then overall the total cell biomass is exergonic. So everything is beginning to stack up, and there's been quite a lot of work. I'm not going to talk about it now, but can later on if you want. In the last few years, showing that if you start with hydrogen and CO2 at high pressure and have hydrothermal minerals, iron-sulfur minerals, you are able to produce carboxylic acids, things like pyruvate, through those reactions. So I say, if you put hydrogen and CO2 though, and in a test tube and shake them up, then they don't react very easily. They're both pretty stable. There is a strong kinetic barrier to their reaction, and it's really, this is the geological pathway to form methane, which is formed under geological conditions, starting from CO2. This is, again, the delta G, positive values here. So this is formaldehyde, and it's quite strongly endogonic to get up to formaldehyde. After that, it's exergonic all the way down to methane, and most of life is around here in this redox state around here. So you get a small amount of energy from not making methane, but from just making these organic molecules around here. What the methanogens specifically are doing is lowering this barrier to the reaction by using a proton gradient across a membrane. So they are lowering the barrier to the reaction between hydrogen and CO2, facilitating that reaction happening. And now the question is, well, how on earth do they do that? Well, this is a first stab at a simple form of the answer. the reduction potential of both hydrogen and CO2 depends on pH. And so this is reduction potential is getting more negative as we go up the y-axis here. So the hydrogen proton couple is not reducing enough to reduce CO2 through to formaldehyde. It's an uphill reaction, just to keep it simple, which is why it doesn't happen spontaneously. That's what you've just seen in the other form in the previous slide. It's basically this uphill barrier there. But that's at pH 7. If we consider the reduction potential at pH 11, where hydrogen is found in these alkaline hydrothermal fluids, it's much more reducing. The reason it's more reducing is that if it passes its electrons onto something else, you leave protons behind. In alkaline conditions, they'll react immediately with the hydroxide ions to form water. And so they're far more likely to do that in alkaline conditions than they are in acid conditions. It's just the Nernst equation. So it's about 59 millivolts per pH unit. Same applies to CO2. If it picks up electrons, it needs to balance charges. If it's in an acidic environment, it'll pick up protons as well. And therefore, again, according to the Nernst equation, it's easier to reduce CO2 to organic molecules in acidic conditions where you can balance the charges with protons. So it's quite simple, but it means that you go from a balanced pH where it's borderline impossible to reduce CO2 with hydrogen to a situation in hydrothermal vents where hydrogen is much more reducing and CO2 is much more easily reduced, so long as you do not have mixing and the pH is kept separate, which means there has to be some kind of barrier to frustrate the mixing. So how is this actually achieved in life? Well, this is work from the last 10 years or so from Rolf Tauer and Wolfgang Buckel, looking at methanogens, how methanogens operate. And again, I'm going to simplify this, but what they're doing is what's called electron bifurcation. They're taking the 2 electrons from hydrogen, they're entering a hydrogenase here, and bifurcation is effectively, I can't say I understand it particularly well, actually, but effectively what it's doing is driving the endogonic reduction of ferredoxin by coupling it to the exogonic reduction of a heterodisulfide to a thiol. So the coupling of the endogonic to the exogonic reaction, and it depends on crossed over reaction kinetics, which is a bit that I don't quite follow, allows the endogonic reduction of ferredoxin. That reduces CO2 through to a methyl group in the end, attached to a cofactor. The methyl groups then pass to a different cofactor, picks up the electrons from the thiol, and it's released as methane. The only thing that's conserved in this process, which can just go rolling on and on and on, is a proton gradient across a membrane, or in fact a sodium gradient, but we'll stick with proton gradients for reasons that, again, I can discuss later on if you're interested. So the reaction of hydrogen and CO2 to produce methane as a waste product, the only thing that's conserved is an ion gradient across a membrane. And the organic itself is a waste product and is lost from the system. So it's the energy change from the transfer of a methyl group from one cofactor to another, which is sufficient to pump 1 proton. Here is here's their wiring diagram of this, and this is based on an E prime, which is to say the typical concentration of hydrogen in the kind of environment where methanogens grow, which would be only around 10 pascals, and the reduction potential of ferredoxin in cells, which is 99% reduced in cells. That's a long way from equilibrium. At equilibrium, it would normally be almost entirely oxidized. The fact that it's almost entirely reduced means you're far from equilibrium. So the E prime here is minus 300 millivolts, and yet hydrogen at minus 300 millivolts is reducing ferredoxin at minus 500 millivolts. So there's a 200 millivolt difference, which electron bifurcation is overcoming. So what's it using? Again, we're seeing this same 200 millivolt difference, and here's what's happening. Here's this membrane protein, the energy converting hydrogenase. It's proton motive, which means the protons are flowing through it. It's taking the electrons from hydrogen and it's passing them on to ferredoxin. So it's reducing ferredoxin. And the ferredoxin is reducing CO2, using ATP as well, not to make a waste product this time, but to drive carbon metabolism. So this is, again, the wiring diagram from Tauer, and you can see, I hope, that it's basically exactly the same. It's minus 300 millivolts is hydrogen under those conditions, minus 500 millivolts for ferredoxin. And now it's the proton or sodium gradient entering here, which is driving that 200 millivolts difference, enabling hydrogen to reduce CO2 via ferredoxin. So we don't need to do electron bifurcation and pumping at the origin of life if we have a natural proton gradient. So the first question from this is, well, all we really need to do is drive the ECH, the energy converting hydrogenase. ATP synthase, I said earlier on, we can put it aside as being a product of evolution and natural selection, and we don't actually need ATP necessarily at this early stage of evolution. There are other ways of doing it, which I can talk about later on as well. So let's focus on the energy converting hydrogenase. Can a natural proton gradient in a hydrothermal vent drive the work required to fix CO2 or any other work? So we modeled this. I won't go through the equations. I've been asked specifically not to go through equations, but it's very simple conceptually. We have an inorganic barrier in a vent. We have this cell wedged precariously in the barrier. This is not intended to be realistic. It's simply intended to make it mathematically more tractable. So 50% of this cell is exposed to the acidic laminar flow in an ocean channel, if you like, on one side, and 50% is exposed to alkaline hydrothermal flow on the other side. And we have a membrane that we can, in the model, we can specify the permeability of the membrane to protons, and we have some proteins inserted in the membrane. We can specify in the model the proportion of the surface area which is composed of those, and the rate of proton flux through these membrane proteins, and the rate of proton flux through the rest of the membrane. We can calculate whether or not it will power work. And the answer is, if you have a modern membrane with a permeability equivalent to bacteria or archaea, then the proton gradient collapses immediately. In effect, the protons come rushing in through the proteins, and we don't have any pumps. So the gradient collapses immediately, and the proton concentration and the charges will balance between the inside and the outside within a second in the model itself. And it doesn't matter how big the pH gradient is, it's going to collapse within seconds. But if that's with a modern membrane, if we have a membrane which is very permeable to protons, like a fatty acid membrane, for example, here's what we've just seen. So this is a modern membrane, and then this is orders of magnitude less permeability. So this is 1 order of magnitude less permeable down to about 5 orders of magnitude less permeable, which is equivalent to a fatty acid vesicle. And here protons can come in, and then they're able to leave again passively, or we're assuming as well that hydroxide, the passage of a proton is equivalent to a hydroxide ion going in the opposite direction or coming in the same direction. So we're assuming equal permeability, and what that means is that you're not collapsing the flow because the rate of removal of protons is equivalent to the rate at which they're entering. So you find a balance, a long-term balance, which is in fact conserving almost all the energy in that system. So if you have a leaky membrane, you don't need a pump. You can rely on the hydrothermal flow effectively to keep neutralizing the protons, so that any protons that come in get neutralized by hydroxide ions and washed away as water. So you can maintain the gradient by hydrothermal flow. And what that means is if you have protocells with a very leaky membrane, you can power work effectively as methanogens do. This is all in silico. So you could power an ECH and an ATP synthase just by a geologically sustained proton gradient, so long as you have very leaky membranes. Now, I won't go into any detail on this, but the key protein here is the energy converting hydrogenase that I mentioned already. I mentioned electron bifurcation. Methanogens and acetogens do it in a different way. Their wiring is a bit different. This is acetogens in the middle, and this is roughly what I've shown you already with the hydrogenase reducing the ferredoxin and the heterodisulfide. So we have an influx of protons through the ECH, reducing ferredoxin, and the electron bifurcation is pumping protons out again. So then we have the circuit of protons coming in and leaving. So this is just pumping. So this is what you've seen already. It's exactly equivalent to this, and the flux of protons is coming in. What acetogens do, bacteria, the other domain of life, is they've inverted the flux and they're now pumping protons out. So they are oxidizing the ferredoxin. They've wired up the electron bifurcation slightly differently, and they're using only the ATPase and the NADH from the electron bifurcation to reduce CO2. So I'm going to put this aside. This is simply, seems to me to be the deepest divergence is about the direction, the polarity of the energy converting hydrogenase. Are protons coming in or are they going out? This is about pumping. And we've said already that in the absence of pumps, this system should work. And we've now simplified it down to a protocell with a leaky membrane and one protein in the membrane, which can reduce ferredoxin. That the structure of this we know from a couple of years ago. It is actually, these are the core subunits of complex 1 that we have in our own mitochondria. The energy converting hydrogenase, you may never have heard of it, but it's this is exactly the same as complex 1, which has additional subunits in the membrane and additional subunits in the matrix arm. And you can see as well that this is an iron-sulfur clusters and a nickel cluster here. And again, the distances are suitable for electron tunneling, for quantum tunneling within these clusters. Now, I've talked about pH, and it seems that 2 of these clusters are unusual in that their redox potential is controlled by pH. This is not typical of iron sulfide proteins, but it is typical of these iron sulfur clusters here. And this was work from some years ago now, looking at the reduction potential of these clusters at different pH. And you can see that again, it's more or less following the Nernstian, it's minus 50 millivolts roughly per pH unit. So that the more alkaline the 2 4Fe4S clusters are that are next closest to this pore, the more alkaline they are, the more reducing they are, and the more acidic they are, the more easily reduced they are. So again, we have a simple potential switch here, which I've tried to summarize in this. So how I imagine it, this would be my working hypothesis, if you like, for the origin of life, is we have a proton pore in the membrane. And if protons come through and bind to amino acids in the vicinity of these 2 4Fe4S clusters here, then it makes them relatively easy to reduce. The nickel is involved in picking up the 2 electrons from hydrogen, and I'm assuming that quantum tunneling would modulate the reduction potential, as would protons binding to these sites here. So it's relatively easy to reduce these iron sulfur clusters when protons are bound. When the channel closes and the protons detach, it now becomes much more reducing and is now in a position where, so we know that this is a minimum of 200 millivolts difference because we know that this reduction takes place, and it's now reducing ferredoxin, which it couldn't do under the conditions where hydrogen can reduce that. So it looks as if it's a simple redox switch controlled by pH, whether or not protons are bound. And this is, again, pH is a bulk property of solution, of proton concentration in solution. What we're actually dealing with is a channel with fixed water, with protons bound to certain fixed waters or to particular amino acid residues. And so the pH, again, I value your help on how to conceptualize this, but it seems to me that there's a binary switch. Either there's a proton there, in which case it's effectively pH 0, or there isn't a proton there, in which case it's effectively pH 14. In other words, you've got far more electrical potential shift depending on the presence or absence of a proton in the vicinity of those. So the amount of power that you have available is potentially quite large. And again, I'm not a physicist, and so I'm out of my depth on some of these questions. I'm trying to set up the possibility, which I would appreciate discussing with you later on. Unlike the ATP synthase or a lot of these proton pumps, it's quite easy to imagine an abiotic or prebiotic version of how this might work. So what I'm showing here are iron sulfur clusters bound to short non-coding peptides, could be as short as 5 or 6 amino acids, possibly even shorter than that. I've got the same number of FES clusters here. I've got some kind of a transmembrane peptide. Again, there are some very simple ones. Gramicidin is the simplest proton pore that we know about, but really anything that spans the membrane and contains fixed water should allow protons straight through. So again, there are issues about charge, which will be interesting, but in principle, this system could work. Again, you'd need protons binding sometimes and coming off other times, but all that requires is variations in proton flux in the environment, whether or not there happens to be a proton coming through right now or not. So in principle, the proto-ECH doesn't have to have any more in it than a few amino acids and some iron-sulfur clusters. And it should be able to do that job of reducing CO2. So we've again done some modeling on this. This was a few years ago now, and I won't go through this in detail, but this is similar setup to what you saw before. We've got a protocell wedged precariously in the membrane. On one side, it's acidic, pH 5 to 7, we can vary that. We've got ferrous iron. On the other side, it's alkaline, pH 9 or 10, and we've got bisulfide ions there. They can enter the cell across the membrane, and they can form iron-sulfur minerals. Those minerals could interact with amino acids or small peptides, organics, basically. And if they do so, we assume that there's a proportion of them might go to the membrane, and if they're in the membrane, would be able, like an ECH, to drive CO2 reduction in the same way that I've been talking about just now. To make, in broad terms, organics, amino acids, I showed you thermodynamically, are the most likely to be formed, and fatty acids. So we're assuming an initial bifurcation when we're making amino acids, which will interact with these iron-sulfur minerals, and fatty acids, which will go to the membrane, driving growth. So the amino acids interacting with the iron-sulfur minerals going to the membrane will be inherited as part of the membrane. So this is a type of membrane heredity, and therefore any daughter cells would tend to also be picking up the amino acids and the machinery in the membrane capable of CO2 reduction. And we could show that under some conditions that would indeed drive growth. So you can see down at the bottom here that cells grow, then divide in half, grow, divide in half. Protocells actually do this kind of thing. They're very labile. They fuse together, they separate again. It's not difficult for fatty acid vesicles to more or less just divide in half. But it raises some immediate questions about, are any of these conditions at all reasonable, or are we just imagining the whole thing? And so this leads into some experiments, and Hannity, who's here, has been instrumental in doing a lot of this work. So the literature says that these alkaline hydrothermal vents are too harsh for these vesicles to survive in. So we looked at a range of pH between 5 and 12, temperatures between 50 and 100 degrees, which is roughly what you'd normally find in these vents. Salinity, we assumed ocean salinity. It's uncertain 4 billion years ago exactly how saline the oceans were, but it's a conservative guess to say they were just as saline as they were today, if not more so. And divalent cations, which can play havoc with fatty acid vesicles, magnesium and calcium, again, a modern ocean conditions. Most work, in fact, virtually all work until now, says that if you have conditions like this, you'll never get vesicles forming. But they use very simple systems with a single fatty acid. So what we did was look at the range of fatty acids and fatty alcohols that have been formed under these kind of conditions. And we used a selection of 12 of the most common ones. This is decanoic acid alone. And you can see this is the steep region here is where you're forming vesicles. Down here, you've got effectively got micelles, which are basically tiny and dissolved. And over here, you've got globules of fat. So you get vesicles with a bilayer membrane around here, and it's just this area around pH 7 where you form them. But if you have simple mixtures of fatty acids, they're stable up to around pH 9. And if you have 5:1 ratio of fatty acids to fatty alcohols, then we're stable up to about pH 10. If we have a 1:1 mixture of these things, then we see this messy graph here, which seems to imply that you get fatty acid vesicles at all pHs right up to 13 and down to 7. And that is in fact the case. These are vesicles. This is by cryo-EM, and this is just confocal microscopy. pH 12, 70 degrees centigrade, modern ocean salinity and calcium and magnesium concentrations, same again here, pH 12. So these are bilayer vesicles, or protocells, we like to call them, forming under alkaline hydrothermal conditions without any problem at all. In fact, it turned out that you need alkaline hydrothermal conditions to make them form at all, because you need to dissolve the fatty acids in the first place. They need to be deprotonated to form these membranes. The other question, which we've just finished writing up, is, well, can we make 4Fe, 4S clusters under these kind of conditions? And the answer is yes, amazingly simply, just with cysteine alone at pH 9, where it deprotonates. This is the classic shoulder that you get on UV-vis spectroscopy, showing an iron-sulfur cluster. We've subtracted the baseline here. These are different concentrations of cysteine, up to 5 millimolar cysteine and down to 0.1 millimolar cysteine. So we're getting 4Fe, 4S clusters by UV-vis spectroscopy down to less than 1 millimolar of cysteine, about 800 micromolar cysteine. We can see them by Mossbauer spectroscopy. Quite interesting differences between 3.5 millimolar cysteine. The iron and the sulfide are kept stable at 1 millimolar in these cases, up to 10 millimolar cysteine. So at 10 millimolar, we're seeing mostly mononuclear clusters and 2Fe, 2S clusters. And at 3.5 millimolar, we're seeing a lot more 4Fe, 4S clusters as well as these others. And by cyclic voltammetry, we're getting reduction potentials in the range of modern ferredoxins using only cysteine as a ligand. So not even polypeptides in this case, and quite low concentrations of cysteine, down as little as 0.8 millimolar of cysteine as well, so long as you keep that ratio roughly the same. So it seems to be surprisingly easy to form both the protocells themselves and the iron-sulfur clusters that we need for this kind of arrangement. We've done more modeling. I won't spend long on this, but this is what you've seen already. Just thinking about catalytic feedbacks. So metabolism is actually branching. To make nucleotides, to get to a genetic code, we would need to have, they're made from amino acids, from sugars. You require an energy currency, something like acetyl-CoA would do, and you can make nucleotides from that. So before you can have a genetic code, we have to make nucleotides. For protocells to get better at making nucleotides, there has to be some way of, well, improving yourself at making nucleotides, and that implies positive feedbacks, which implies catalysis. And so we looked at all the different forms of catalysis we could think of, just theoretically, first of all. So feeding back on CO2 fixation, or feeding back on any of these other pathways, or feeding back on CO2 fixation and itself as autocatalysis. And what we can see across here is that because you're branching flux, if you have a low catalytic rate, then you're forming fewer organics. But as soon as you pass a threshold, that's a catalytic threshold of feeding back here onto CO2 fixation, it takes off. In other words, nucleotides will remain in the system just for thermodynamic reasons and catalytic reasons because of CO2 fixation. Interestingly, autocatalytic feedback doesn't work, doesn't help. It helps a little bit, but effectively what we see here is that if you start accumulating nucleotides, the rate of protocell growth slows down. So the faster rates of growth are associated with some autocatalysis, but not much. So we're in this corner over here. So what that says is that it's quite robust, and you don't have a parasitic takeover, parasitic collapse from nucleotide autocatalysis, nucleotides copying themselves under those conditions. Okay, so all of that is really to say that kind of metabolic heredity can take you so far in protocells. It won't get you to a code, but it gets you from having a very simple system to having something that we would recognize as modern metabolism. And a lot of this has now been achieved under these conditions, under prebiotic conditions. Only nucleotide synthesis has not yet been achieved. And there's a couple of people in my lab who are working on exactly that, and they've done a lot of the steps now. So it's something which I think is going to happen in the next couple of years. Right, so I want to take a step back again, and probably for the last 10 minutes or so now, think about what actually can be happening in a prebiotic system. So I'm taking it for granted in the previous slide here that we are making organics across a protocell membrane. But before we get to that protocell membrane, we're dealing with the inorganic barrier. And I'm looking for homology, really, between an inorganic barrier in a vent and a protocell. So if we have this is roughly what you've seen here. We have a channel alkaline pH with hydrogen in it. It's more likely to reduce the barrier to produce protons in the alkaline channel. Those electrons can cross the barrier, in theory, to reduce CO2 this is not intended to be formaldehyde, this is just generally organic molecules on the other side. So we have an electron flow across this conductance across this barrier, making organics on the acidic side of the barrier. If you follow that through to a logical conclusion of making protocells, then the protocells would be here. What they will be receiving would be electrons from the barrier, and the protocells would be lining the acidic side of the channels. We didn't find that our protocells were stable under acidic conditions. They were only stable under alkaline conditions. The head groups protonate under acidic conditions. It's possible to imagine that you form organics and that they wash away and go somewhere else in the system and accumulate elsewhere in the system. But this is also not consistent with life insofar as cells take hydrogen inside the cell and it's protons which are flowing into the cell. So this seems to be inconsistent in its topology with what I would like it to be doing if it were a modern cell. Here is the alternative. Whoops, sorry, lost it. Here's the alternative, which is we have protons crossing this barrier, and the CO2 reduction is taking place right on the very edge of the alkaline side. We still have hydrogen in alkaline conditions. We still have CO2 in relatively acidic conditions because protons are crossing the barrier, and we're making organics right at the edge here, which means we'll be forming protocells on the alkaline side. And this seems to be, to me, to be topologically more equivalent to a modern cell than that does. But it's an interesting question. Can we do the experiments? Now, a former student of mine, Victor Soho, just succeeded in doing this with Ruben Hudson, which was published in PNAS a couple of months ago. And he's shown really beautifully, this is the setup here. This is on a microfluidic chip, and the interpretation that they think is most reasonable is that hydrogen is giving up electrons to the barrier, producing protons to form water. The electrons are crossing the barrier and reducing CO2 on the acidic side. What they've shown in these NMR using deuterium, deuterated water, and 13C is consistent with this, which is to say the protons are coming from the acidic side, and the CO2 is definitely the organics, the formate in this case is definitely coming from CO2. It's not a contamination. And then on the right hand side are other possible ways of seeing it, most of which do not correspond to what we're actually seeing over here. I'd like to bring your attention to D. This is consistent with their results, but seems more problematic, at least to the authors. I was one of the authors. I wouldn't disagree with the conclusions, but what this is showing is protons crossing the barrier and CO2 crossing the barrier, as I've just suggested to you, forming on this side organics, formate in this case, but with the protons deriving from over here and the carbon dioxide deriving from over there. So in terms of the in terms of the labeling, the isotopic labeling, this is consistent with the picture they're seeing, but it begs the question, can protons and CO2 cross this barrier? This is something which has been put forward long ago by Mike Russell again. I mentioned him earlier on. This was an earlier depiction from Mike Russell of essentially the same thing. He's got protons crossing the barrier. He's also got electrons from hydrogen crossing part of the barrier as well, and he has them meeting in the middle. Molybdenum is potentially important because molybdenum will do 2 electron transfers, whereas iron only does 1. Nickel will also do 2 electron transfers. He's forming formate inside the barrier. He calls it a membrane, being a geologist, but this is 4 micrometers in thickness. This is from his work in JPL. But still, it's not really known, will protons cross this kind of barrier? We've done some of our own microfluidic work, And this is the barrier, a thick barrier in this case, precipitated in the mic. This is a relatively thin one and a much thicker one after a few hours inside the microfluidic chip. You can see here that we're getting iron sulfide minerals like McKinowite. This is the structure of McKinowite that you're looking at by EM. And this is just looking at proton pH gradients using just universal indicator initially. Sorry, I'm running out of voice here. And you can see that even in the absence of a barrier, the laminar flow retains pretty much fairly sharp pH gradients across this. And when we have a barrier here, the barrier is quite diffuse. And what we were immediately struck by is that the barrier looks acidic. And we look more carefully at that. Here's another example. So this is the alkaline channel in blue. The indicator doesn't actually give us a very good indication of what the pH actually is. It's somewhere between 8 and 11, and we were infusing it at pH 11. And the acidic flow was pH 5. And you can see that this is the barrier here. This is the acidic flow. This is the barrier. Some aqueous spaces in the barrier. And right here, it's still pretty acidic. And you can see that difference. This is a 70 micrometers across there. And you can see that it's acidic. So we did some calculations, and it's a 5 pH unit gap across that 70 micrometers. And we did some stop flow experiments, and it showed that the proton flux is 2 million times faster than the hydroxide flux in crossing these barriers. I won't go through those results now, which shows that we could have potentially 3 pH units difference across a single 25 nanometer nanocrystal of McKinowite. And this is the final slide then. This is what I just showed you a moment ago, tilted through 90 degrees. On the outside, we have the protons. On the inside, it's relatively alkaline. And this is the prebiotic system that I was talking about. Here is roughly what we'd be thinking about in a barrier itself, if what I've just said is remotely correct. And the topology is not quite right. It's basically still the same as the topology of electrons crossing the whole barrier. We would need, on opposite sides of single crystals, quite steep pH gradients, so that on one side we either have protons binding or we don't have them binding. That would modulate the reduction potential of the crystal itself. It's been shown that that does indeed happen, which makes it easier to pass electrons from hydrogen onto the crystal itself and then off again at the other side onto CO2 to make formate and organic molecules. I've been trying to think of a way of making the topology work more like the energy converting hydrogenase. It's not easy to see exactly how this would work. All I can say for sure, or relatively sure, is that it's perfectly reasonable to have 1,000 times higher proton concentration here than it is over here. And that the electrons definitely are coming off hydrogen onto the McKinowite crystals themselves, nanocrystals, and can reduce CO2 to formate at least, and probably more that way. What's happening in terms of mixing in these spaces, whether CO2 can cross this, I really don't know, and I would appreciate if anybody has any views on that kind of thing. That's it. I'm going to stop there with those questions. This is the lab, and I feel obliged as ever to say that none of this experimentation has been done by me. It's been done by the people in the lab, and it's also it's pretty brave to do a PhD or to work as a postdoc in this field, because it's quite an angry field. People get very committed to their way of seeing it, and there's no clear career prospects at the end of it. You've got to work for it. It requires bravery to come into this area at all. It requires some bravery, I think, to work with someone like me. So I'm very, very grateful to them for venturing into this field and doing these experiments. We've come a tremendous way, I would say, over the last 5 years. Thank you. I'll stop there.
Nick,